In this video lecture, I will be talking on mitogens. Mitogens are substances that induce mitosis. In case of T-dependent antigen, where it requires T and B cells interaction, only a small fraction, 1 in 1,000 or 1 in 10,000 of T cells are able to recognize the antigen and become activated. However, in T independent antigen, where the T and B interaction is not required, and this interaction or this T independent antigens or this T independent antigen is known as mitogen, which induces cell division in high percentage of T and B cells and upregulate antibody production in the absence of T cells. Examples of mitogens. The first example is sugar binding proteins known as lectins, which bind glycoproteins on surface of lymphocytes. Examples of lectins are concanavalin A or CoA. The source is jack beans, which has a capacity to activate T cell. Phytohemagglutinin, and the source is kidney beans, and it has the capacity to activate T cell. Buckwheat mitogen, the source is buckwheat, and it has the capacity to activate both T and B cells in the absence of T lymphocytes. The second examples of mitogen is lipopolysaccharides or LPS, and LPS is a component of the gram-negative bacterial cell wall. Functions as B cell mitogen. The third example of mitogens are superantigens, which are antigens that polyclonally activate a large fraction of the T cell, up to 25%. And as can be seen here, this is an interaction between the T helper 2 and B lymphocytes. So superantigens, simply they just connect the T cell receptor to measure histocompatibility. So superantigens bind to residues in variable domain of T cell receptor and to residues in class 2 molecules outside the antigen binding cleft. So it has a capacity, the superantigen, to produce an effective immune response without or outside the antigen binding cleft. Examples of superantigens include staphylococcal enterotoxins, this is called poisoning; toxic shock syndrome toxin 1, produced by staphylococcal aureus, and these superantigens lead to hyperactivation of the immune system and release of cytokines by activated T cells. Thank you very much Professor Ahmed Bola.